innate immunity. And coming to the cells, okay, cells, different types of immune cells are there in our body. We will discuss later what are they. In this branch of innate immunity, uh, phagocytes are involved actually. Okay, polymononuclear cells and macrophages are acts as the phagocytes. And natural killer cells, mast cells, dendritic cells. I will tell you the detailed information later about each and every. Will talk. I will talk in each and about each and every cell. And especially in this particular innate immunity, these are the cells actively involved. And uh, what are the what are the striking features of innate immunity? Okay, innate immunity is a particular barriers are there. Okay, one or two to three barriers are there which protects with barriers are the protectors, protecting walls. Whenever the foreign invaders come and contact with our body surface surroundings, it cannot allow into the passage through its barriers. Okay, those barriers are physical barriers, that may be physical barrier or mechanical barrier, and cellular barriers are also there, and chemical barriers also be there. So there are different types of barriers are there. We will discuss one by one. And barriers, okay, in another way we can call also a anatomical barrier. Okay, uh, one is that anatomical barrier, second one is that physiological barrier, third one is that phagocytic barrier, fourth one is that inflammatory barrier. Okay, these are the four types of barriers are there. Coming to the anatomical barrier, okay, anatomical barrier immediately it will protect the body. For example, coming to the anatomical barrier example, skin is one of the anatomical barrier. Okay, um, stratum corneum, in stratum corneum is the outermost layer, and which prevents the entry of the bacteria virus. Okay, stratum corneum is the outermost layer. Stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, stratum basal is the five layers of the five layers are there in our skin. Okay, among the five layers, our outermost skin layer is that stratum corneum is the one of the most important uh, anatomical barrier, we can call as physical barrier. Okay, and this is one of the barrier. And second one is that sebum in the skin. Sebum is secreted and it maintain acidic condition. Okay, sebum maintain acidic condition in which some of the bacteria, virus, fungi cannot be entered into our body through the skin. And keratinocytes, okay, keratinocytes also be there in the skin. So keratinocytes also there in the skin, and in which pattern recognition receptors, pattern recognition receptors are there and binds with phagocytic cells. Okay, and these cells are binds with phagocytic cells. Whenever uh, these keratinocytes encountered by the different types of parasites and pathogens, it directly connect with the phagocytic cells. Then phagocytosis will be activated after the contacting with between the karyocyte and antigenic particles of the foreign bodies. And next anatomical barrier is that mucous membrane. Okay, mucous membrane is there in different parts of the different. It lines different anatomical uh, parts of our body, which protects the entry of different parasites and pathogens. Especially, it has antibody A. Okay, antibody A in the mucus layer protects from the different types of parasites and pathogens. Especially, it's acidity also. Acidity also prevents the uh, uh, development of the different types of pathogens and parasites. And so we can also call it as mucus layer, also call it as anatomical barrier. So two of them are anatomical barriers are there, skin and mucous membrane. And second one is that physiological barrier. And our body temperature also acts as a physiological barrier at which different types of pathogens and parasites cannot be survive actively, and not growth cannot be enhanced at that temperature. And pH, our body pH also acts as a physiological barrier where exactly different types of pathogens and parasites cannot survive. And other secretions of different body parts, example, acidic acid of stomach. Okay, acid of stomach can also act as a physiological barrier. And bile uh, from liver also one of the secretion which prevent the different types of pathogens and parasite development. And serum, okay, sorry, cerumen ear wax, or you can also call it as ear wax. And it repels insects, kills bacteria, traps dust particle in the ear entry, and this is also acts as a physiological barrier. Lysozyme, okay, lysozyme is another physiological barrier in where exactly it is there. It is there in our tears and saliva, especially. These two secretions are having a lysozyme enzyme. It kills bacteria. Okay, whenever the bacteria enters into the eye and mouth, the secretions from the eye and secretion from the mouth, tongue, different salivary gland secretions kills bacteria. Okay, these are the barriers, physiological barriers. The nasal hairs, we have nasal hairs in the nostrils, which also prevent the entry of the different dust particles, foreign uh, bodies like bacteria and fungi at different types of entries, infections. Urine also acts as a physiological barrier sometimes. It washes microbes from urethra. Flow of the urine washes the microbes from the urethra and also acts as a physiological barrier. And vaginal secretions sometimes, especially it has acidic condition. In this condition, bacteria cannot grow in this condition. Okay, bacterial growth can be inhibited by the acidic condition of vaginal secretion. So these all of these acts as a physiological barrier. Another physiological barrier is that sebum. Okay, sebum secretes from the sebaceous glands of skin, which also acts as a physiological barrier. These all of these are innate branch of immunity barriers. Okay, this is about second barrier, physiological barrier. And third barrier is that phagocytic barrier. A phagocytic barrier means phagocytosis. Through the phagocytosis, different types of cells, okay, different types of phagocytic cells um, at, get attached with the different types of antigens. And whenever these phagocytes are attached to the antigens on the surface of the pathogens and parasites, get activated after the attachment of the binding to the antigen particle, and get activated. And after the activation, it ingests the foreign body. Okay, attachment is the first step in the phagocytosis. Second step is that ingestion, and third step is that fusion with the lysosome. Whenever the foreign body enters into the cell, that is phagocytic cell, after entering into the cell, the phagosome get attached with the lysosome. Okay, we can call it as phagosome. A foreign body also called as phagosome. After entering into the body, it directly attaches with the lysosome, phagolysosome. The binding of the phagosome with the lysosome is called as phagolysosome combinedly. And after the combined with the lysosome, secretions of the lysosome um, will digest the entire body at a foreign body, and it the pieces, okay, the part of the foreign body will be act as a antigen, especially different types of uh, protein particles, um, different types of lipid particle on the surface of the plasma membrane of the foreign body act as the antigen. Those antigenic part can be digested and presented, and this antigen body goes and binds with the MSC molecule in the cell cytoplasm, and this MSC molecule get collected. These antigen get collected by the MSC molecule and presented on the surface. MSC molecule moves towards the plasma membrane and uh, present the antigen to the other cell. Okay, these especially phagocytic cells present the antigen to other cells. Example, it presents the antigens to the TH cell. It presents the antigens to the different types of the immune cells. Okay, this is another barrier. Okay, one, two, three barriers are there. If antigen just contact with the skin, it eliminated or it destructed by the different types of the secretions, and which if the foreign particle entered into the physiological openings, okay, body openings, and there is the secretions are there. That is the second barrier. If it exempted or escaped from the second barrier, third barrier is it will be encountered by the third barrier, that is phagocytosis. If it is it is excused or and escaped, maybe it may be escaped by the third barrier, and final barrier is there, that is inflammatory barrier. Okay, and defensive response of the body to tissue damages. Okay, the symptoms is that first step is in the inflammation is that redness. Okay, next pain, and third is heat and swelling, and these are the steps are these are the symptoms, not steps exactly. And events means steps. Vasodilation is the one of the step in the inflammation, making it red, warm, swelling. Okay, different types of the uh, pharmacological agents secretion leads to this type of the vasodilation, especially uh, bradykinin. Okay, bradykinin is one of the pharmacological ingredient secreted by the mast cell basophils. Okay, whenever this bradykinin histamine secreted from these cells, whenever this cell get activated, this type of degranulation will be seen. After the degranulation, different types of primary and secondary particles, pharmacological particles will be released, and these pharmacological particles acts upon the different types of tissues, leads to the redness of the skin and redness of the area, infected area, and swelling of the infected area, and warm at the infected area, pain especially due to the bradykinin secretion. It stimulates the pain receptors in the skin. Okay, bradykinin goes and binds with the pain receptors. Okay, and leads to the pain. Okay, leads to the pain, and different types of defense mechanisms are there, especially among the along with these barriers. Different types of defense mechanisms also be there in the innate immunity. Those are complement system. Fever is one of the mechanism, and cytokines secretion also one of the another defense mechanism in the innate immunity branch, in the branch of innate immunity. So this is about innate immunity. If you have any doubts, you can ask now about innate immunity. Any doubts?
No doubt, sir.
Okay, this is about innate immunity. And coming to the uh, acquired immunity. Okay, acquired immunity is another branch of immunity. You may have any idea about this, but I will tell you what exactly acquired immunity is that. Yes, acquired immunity is adaptive, also called as adaptive immunity, and specific immunity uh, against different types of foreign invaders. So in this immunity, we have another two types of subtypes of immunity. One is that humoral immunity. Another one is that uh, cell-mediated immunity. One is that humoral immunity. Another one is that cell-mediated immunity. So coming to the acquired immunity, acquired immunity is developed after the infection, okay, and slow to start, highly specific, and memory will be there in this type of adaptive immunity. And cells, coming to the cells, and two types of cells are actually involved in the adaptive immunity. Those are T lymphocytes and B lymphocytes are actively participate in this type of immunity. And uh, antibodies, okay, antibodies and cytokines are actively involved in this type of immunity. Yes, I will explain one by one. In this presentation, all information tell you about cells and organs. I'll later explain about the cells, but let me complete about acquired immunity. So in this branch we have humoral immunity and cell-mediated immunity. What is humoral immunity? Humoral immunity is the immunity in which the antibodies are actively participated. Antibodies are actively involved in this type of humoral immunity. Where exactly antibodies are, where from where exactly antibodies are produced? Antibodies are produced from B cells, B lymphocytes. Okay, B lymphocytes especially. Whenever the antigens or these B lymphocytes get attached with the antigen and bind with the antigen, and this binding of the antigen to the B cell facilitates the activation of the B cell, and this B cell get proliferated, and one cell it get divided into two cells. One is that memory cell, another one is that uh, plasma cell, and this plasma cell will release the antibodies. Exactly, plasma cell will release the antibodies, and these antibodies are the major play a major role in the humoral immunity, which is the sub branch of the acquired immunity. Okay, in the secondary lymphoid organs, an antigen binds with the Fab region and the end of the body antibody. I will explain what is exactly anti-Fab region. And these cells process the and present the antigens to the TH cell. Okay, and these B cells, okay, B cells get interact with the antigen, okay, and uh, proliferate into the memory cell and effector cell, that is plasma cell. Sometimes these B cells also acts as a professional antigen presenting cell. It presents the antigens to the T cell, that means especially TH cell. And what happens after the presenting the antigens to the TH cell by the B cell? TH cell is sensitized and activated and release interleukins. Okay, the interleukins will be released after the contact with the B cell to TH cell with the antigen. Interleukins helps to the proliferation and differentiation of the B cells. Okay, just listen carefully. This release the interleukins. Where exactly interleukins released from T cells, especially TH cell releases the interleukins after binding of B cells. After binding of the B cells with the TH cell, this TH cell gets sensitized and differentiate and releases the interleukins. And these interleukins goes and binds with the B cell, okay, which is attached with the TH cell. And after the getting information, after the contact with the interleukins on the surface of the receptors, B cells uh, proliferate, differentiate into the two cells. Okay, this is actual story what we have been talking about. And B cells activation will be started after the getting activated. This cells gives two cells. One is a plasma cell, another one is memory cell. And plasma cell will sensitize, release, and synthesize antibodies, especially memory cell cannot really releases any antibodies, but some antibodies attached to the memory cell. But plasma cell always releases the antibodies from its surface. Okay, antibodies. What are the antibodies? Antibodies are the immunoglobulin molecules. It neutralizes the antigen as well as activate the phagocytosis or phagocytic cells and complement proteins. That is different types of, okay, the different types of uh, immunological mechanisms. Whenever the antibodies released into our system, our blood or limb, these antibodies go and bind with the different types of molecules and cells, especially whenever this antibody goes and binds with the phagocytic cell, and the phagocytic cell get activated, and phagocytosis of the attached molecule or attached foreign body will be started. And whenever this antibody goes and binds with the complement proteins, okay, it has the receptor to the complement protein, it has a receptor to surface molecules of the different types of phagocytic cells. So these antibodies, release the antibodies, goes and binds with the different types of cells and molecules. If it is binds with the phagocytic cells, phagocytosis is the result. If it is goes and binds with the complement proteins get activated, and different complement system may activation may leads to the pore formation on surface of the foreign body. Okay, sometimes it also our infected cell only. Okay, on the infected cell, and wherever the antibodies attached to the antigens, these complement proteins and goes and binds with the antibody. And after the attachment of the complement protein, it attracts the different types of subunits of the complement protein. And after the different types of the composition, okay, of the complement protein, it makes a pore on the infected cell, or the on the surface of the foreign body like bacteria or fungi or parasites. This is the importance of the humoral immunity. Okay, what is humoral immunity? Humoral immunity is a branch of acquired immunity. It is the important immunity. In this immunity, antibodies are play a key role. These antibodies are released from the B cell, especially plasma cell, and release the antibodies goes and binds with the macrophages, okay, and different types of complement proteins. And by these ways, our immune mechanisms get activated, and infection may be eliminated from our body by the immoral immunity. This is the one of the branch. Understand? Okay,
Yes,
Namu.
sir. Yes, sir.
Coming to the second second type of second subtype of acquired immunity, that is cell mediated immunity. Cell mediated immunity is one of the sub branch of the immunity, and in this type of the name itself also indicating that cells are actively involving in this type of the immunity, especially T lymphocytes are actively involved. Okay, coming to the T lymphocytes are the three types are there. One is that uh, TH cell, okay, T lymphoid helper cell. Another one is the TC cell, T cytotoxic cell. Another one is that T suppressor cell. Okay, three types of cells are there. Okay, subtypes of cells are there, and these cells are actively involving in the uh, cell mediated immunity. Okay, what is cell mediated? In which cells are actually involved, and whenever the antigens coming to the our system, these antigens are collected by the lymph, and these lymph goes and submit these antigens to the different types of the cells in the lymph node. Okay, the action site of the fighting site or burial battle site is that lymph node. In the lymph node, these antigens and different types of the cells interacted, and whenever the foreign molecules or foreign antigens comes, and the lymph node, our antigen presenting cells are there. Example: B cell and dendritic cell and macrophages and different types of cells are acting as a uh, antigen presenting cell. Especially what happens that in the cell mediated immunity we have talked about the importance of the T cell. Okay, T cells cannot recognize the antigen directly, but it requires some other help, some other cell help to recognize the antigen on the surface of the foreign molecule. For example, whenever the foreign body come and contact with our immune cells, immune system, and some of the cells goes and binds with the uh, foreign microbes. Okay? And for example, if the macrophage or dendritic cell goes and binds with the and bacteria, and this bacterial entire bacteria will be ingested by the phagocytic cells. Okay, and this phagocytic cell digests the foreign bacteria body, entire body, and on which on the surface of the body, different areas are there actively. Those areas are specific, and which activate the our immune system. It has a capability to activate the our immune system. Entire the surface cannot having any any other particles among the surface. Those are the specific spots are there, specific areas are there. Those only having the capacity to evoke our immune system or stimulate our immune system. So those areas trimmed out by our lysosome enzymes, and these areas are collected by the MSC molecule and present on the cell surface, especially antigen presenting cells. Okay, then only our T cell goes and binds with those antigens. Okay, and MSC molecules are the two types. Okay, MSC molecule 1, MSC molecule 2, and in this type of the molecules are play a major role in the attachment of the T cell. Okay, differentiation of the difference between MSC1 MSC2 is that always our T cells goes and binds with these MSC molecule. For example, the TH molecule always binds with the MSC molecule 2, and TC molecule, TC receptor always binds with the MSC molecule 1. This is the rule. Okay, MSC 1 molecule binds with the TC cell receptor. MSC molecule 2 always binds with the TH cell receptor. Okay, that is CD4 receptor, and C molecule TC contains CD8 receptor. Okay, this is binding between CD molecule and MSC molecule along with the antigen activates the T cell. That may be either TH cell or either or T cell, and the binding facilitates the activation, the differentiation, and proliferation. After the proliferation, these TH cell, TC cell proliferate into the two types of cell, that is memory cell, effector cell. Okay, effector cell is a specific cell which binds with the, which has the capacity to bind with the three types of cells. Okay, one is that B cell, another one is that macrophage, another one is that uh, TC cell, especially in case of TH cell. What happens uh, after binding of the MSC molecule to with, assume that this is MSC molecule, and this is a T cell receptor, and this is the antigen. Binding facilitates the activation, and after the uh, and also some of the chemicals, and those are the uh, interleukins, the cytokines released from the macrophage or antigen presenting cell. This is antigen presenting cell. Presenting cell. From the antigen presenting cell, some of the interleukins goes and binds with the contact with the TH cell, and TH cell again releases some of the interleukins and goes and binds with the antigen presenting cell. This communication further enhances the activation of the TH cell, which leads to the proliferation of the TH cell into two cells. One is that memory cell, another one is that effector cell. And this effector cell has a capability to bind with the three cells. That is in case of TH cell. Okay. This TH effector cell can bind with the B cell. Okay, get active after binding with the B cell. It can activate the it can has a capability to activate the B cell. Also, it may bind with the macrophage cell. Okay, also activate the macrophage cell. It also bind with the cytotoxic cell. Okay, TC cell, and this effector cell has a capacity to bind with these three types of cells, and whenever the B cell get activated, you know about the humoral activation, and these antibodies goes and binds with the different types of antigen and neutralizes the antigen. And if the TH cell activates the macrophage, it goes and binds with the foreign body, and it engulfs the foreign body. Phagocytosis will be there, will be the consequence. If it is, if it activates the T cell, TC cell, it proliferated, get proliferated into two cells, effector cell and memory cell. And this memory cell goes and binds with the direct if infected cell, and releases the different types of enzymes, especially porphyrins, granzymes. Okay, the most harm enzymes are the effective enzymes, porphyrins. Okay, so second particle is that granzymes. Okay, of the granzymes also released by the our C cell, okay, the cytotoxic T cell, and after releasing of the T cell, it it fights with the it releases whenever the granulozyme, not it can also pronounced as a granulozyme, and this granulozyme goes and binds with the uh, enzymes. Okay, some of the uh, uh, mediators, especially it goes and binds with the caspases. Okay, and these caspases are there in the mitochondria and play a key role in the apoptotic pathway, apoptosis pathway. You know the what is the meaning of the? Did you know the meaning of the apoptosis? What is apoptosis?
Programmed cell death.
Yes, whenever this granulozyme released from the TC cell, and this TC cell granulozyme goes and binds with the go and enter into the infected cell or foreign body, that may be bacteria or parasite, and this enzyme goes and binds with the caspases. Where exactly these caspases are there? In the mitochondria of the cell, that may be the our infected cell or foreign cell. This granulozyme goes and with the caspase and activates the pathway of the apoptosis. After the binding of the granulozyme with the caspase, the further enhancement of the apoptotic pathway is taking place. So this is the way to kill the cell in the TC cell. TC cell will kill the foreign body or our own cell by these two ways. What are they? Making the pores by the help of porphyrin, making the uh, activation of the or cell death, natural death process of the cell by the binding of the granulozyme with the caspases enzyme. By these two ways, TC cell kill the other cell. Okay, this is about the cell-mediated immunity. Okay, and once again, I am giving the uh, one more uh, overview. Humoral immunity is a sub branch of the acquired immunity, which in which antibodies are play a key role. And you know about the antibodies. I will tell you later what are the exactly antibodies are there. And next, in the cell-mediated immunity, different types of cells, and especially T cells, whether maybe TH cell or TC cell, play a key role in the elimination of the different types of foreign invaders. Okay, this is about cell-mediated immunity and acquired immunity, especially, and this is about types of immunity. Okay, coming to the next topic. What is next topic? Next topic is that cells. I would like to talk about cells of immune system. Okay, cells of immune system. We have been talking about different types of cells, but we know something about cells, but I will tell you the detail form. Okay, one by one. And first of all, we uh, I would like to tell you about the innate branch cells. Those are involving in the innate immunity branch of our immunity. Okay, coming to the innate immunity, with different types of cells are actively involved. What we have talked in the previous session, you have uh, this information in the page number 6. Okay, and coming to the different types of cells, all the cells uh, we have uh, myeloid lineage. Okay, myeloid lineage from the stem cells. Different types of all these cells are developed, are synthesized, or manufactured, and granulocytes. Okay, granulocytes are the one of the innate immunity cells, and these cells are frontline cells for the initial response whenever the infection will be there in the body. These granulocytes fight will be there in the front line and first line attacking cell. We can also call as granular cells can also called as first line attacking cells. Okay, what are the granulocytes? Okay, these mast cells, eosinophils, okay, different types of granulocytes are there. Okay, for example, especially three types of cells are there: neutrophils, basophils, eosinophils are the granulocytes. Okay, and coming to the neutrophils, polymorphonuclear cells, we can also call as polymorphonuclear cells due to the presence of multi-lobed nuclei. It contains multi-lobed nuclei. You can see in the image also. Less lifespan, 5 to few days, not 5 exactly, few days. They will die in bloodstream. Okay, natural apoptosis is the process. And coming to the functions, patrolling in the body. Okay, it is main function of the neutrophils is that patrolling property is the function. It patrols the throughout the our body, and it searches for the foreign invaders. Okay, always 24/7, and through the bloodstream in search of microbes and cause early inflammation. The main inflammation causing cells are neutrophils. Okay, and cytoplasm contains two types of granules. That is not necessary to tell you about those granules. And coming to the basophils, another granulocyte site cells, and innate immunity cell, and very rare cells. Okay, very potent at the present time. Okay, whenever these cells are activated, very potent. And non-phagocytic cells, these are the non-phagocytic cells, basophils, and non-phagocytic granulocytes are there in this cells. Granules also there. Large granules with basophilic proteins. Okay, in the granules, what are the contents present? Which contents? Which type of the contents are there? Basic contents. And when they attached with the circulating antibodies, okay, and these antibodies releases into the circulatory system, and this cells basophils goes and with the binds with the antibodies. This binding of the antibody with the basophils facilitates the degranulation. Okay, which with where exactly granules are there? Granules are there in the cell. The binding of antibodies to the basophils facilitates the degranulation. Granules contain histamine and different types of active ingredients. This interhistamine uh, acts upon smooth muscle cells and with muscle contraction, vasodilation. Different properties are there for the histamine pharmacological ingredient. And function of the granulocyte is that anti-parasitic property. Okay, helminth. Whenever the helminth uh, parasites come in contact with our immune system, the most these basophil number will be increased and goes and binds with the goes and fight against the helminth parasites. Okay, and anti-allergic, whenever the anti-allergic reactions are there, and these anti-allergic functions also be taken by this basophil, and it secretes the cytokinin to modulate the B cell and A cell. That is not an important point, but you can remember anti-parasitic property. So this is second type of granulocyte, and third type of granulocyte is that eosinophil, and these are the like neutrophils, motile phagocytic cell, migrate into tissue spaces. And these cells migrates into the tissue spaces. The process of migration is called as diapedesis. From the blood to the tissue spaces. The process of the transportation or migration from the blood to the tissue spaces is called as diapedesis. And bilobed nucleus is there. You know about these all these things. The function of the eosinophil is that it defends against the parasites. Same function. Basophil, eosinophil fight against the helminthic worms. Okay, main function, it makes allergic reaction. Okay, it makes allergic reactions in our the body. It fight against the helminthic worms. This is about the granulocyte. And we have another type of cell, that is mast cell. Okay, mast cell is also one of the another cell. Uh, Before going to tell you about the mast cell, I will show one of the image. Okay, this is the plant or synthesis of the different type of the cell from the hematopoietic stem cell. From one cell, these different type of the cells will be produced from the stem cell, that is hematopoietic cell in our bone marrow. From these hematopoietic stem cell is there in our present in our bone marrow, from which different types of lineages, so different types of immune cells and other blood cells also produce. And this mast cell exactly uh, produced from the myeloid progenitor, okay, released from the bone marrow into the bloodstream. And what is the function of the mast cell? Especially contains FC receptors. Okay, it contains FC receptors for the immunoglobulin E. Okay, it has a receptor for the immunoglobulin E molecule, and whenever the immunoglobulin E molecule antibody goes and binds with the this receptor, it gets sensitized in the at the first time. In the second time, it proliferated into the two type. Furthermore activation, not proliferated, but exactly it furthermore activation will be there. And degranulation is very important point, is that degranulation of the mast cell is taking place after the second time contact with the same allergen. So this FC receptors for the immunoglobulin is there on the mast cell, and after degranulation, histamine will be released. It makes it may leads to the allergy in the person, infected person, affected person. And serotonin, another pharmacologically, we have talked about these molecules in the hypersensitivity reaction. And body bradykinin and leukotriene, prostaglandins, different types of the uh, molecules will be released into our system, makes different types of reactions, effects on our body. And another cell is that mononuclear cells are there. A mononuclear cell, for example, different types of mononuclear cells are there. For example, monocytes or macrophages, mononuclear phagocytes. Okay, we can also called as mononuclear phagocytes, macrophage, monocytes. Where exactly these cells are there? Here. Okay, monocyte. These cells also evolved from the comes from the myeloid progenitor cell, produced from the myeloid progenitor cell. We can monocytes are circulates in the body. Monocytes always circulate in the body, patrols for the different types of agents. Migrate in between tissue spaces. It has a capability to migrate from the blood to tissue spaces. And in the tissue space, where exactly cell differentiate into the macrophage. In the bloodstream, we can call as monocyte. If it goes migrate into the tissue space, we can call as macrophage. Same monocyte differentiate into macrophage. Okay, lysosomes are present in the more number, involving in the removal of particulate matter. Internal scavenger, we can also it can also act as a internal scavenger. And insoluble, it releases insoluble or cellular antigens will be cleared from this phagocyte. Okay, names of the macrophages are the different names are there for the macrophages based upon the site where exactly these macrophages are there. If the macrophages are there in the liver tissue, if the monocytes migrates into the liver tissue surface, it differentiate into the Kupffer cell. Okay, we can call it as a Kupffer cell. in the liver tissue. If it is migrates into lung tissues, we can call it as alveolar cell. Okay, alveolar cell. If the monocyte migrates into the connective tissue and differentiate into histiocytes, if it is migrates into kidney tissue surfaces, it differentiates into the mesangial cells. The mesangial cell. If it migrates into the brain cell tissue, the monocyte migrates into the brain tissue, especially central nervous system. It differentiates into the microglial cells. Okay, these are the all are the macrophages which protects from the different foreign invaders. If it migrates into the bone tissue surface, uh, it differentiates into the osteoclast cell. If it is goes into the synovial fluid, synovial cell. Okay, different types of macrophages are there exactly based upon the tissue surfaces. Okay, different types of the oxygen dependent phagocytosis process is there. Okay, oxygen independent phagocytosis pathway is there. Two types of pathway for the macrophages or any other phagocytic cells, and by these two ways the macrophages clears the foreign bodies from the system, infected system. And we have another cell, that is another mononuclear cell, that is that dendritic cell. Okay, the dendritic cells involved in the antigen presenting or antigen processing. Okay, this cell involving in the processing of the, it processes the antigen, and it presents the antigens to the Th cell or Tc cell. And these dendritic cells are two types, uh, especially based upon the site. Excuse me. Langerhans cell, we can, dendritic cells can be called as a Langerhans cells if it is there in the skin. Langerhans cells can also be called as intestinal cells. Okay, interdigitating cells. Okay, interdigitating cells. Don't bother about the types, but remember this dendritic cell acts as a antigen presenting cell, especially professional. This macrophage and dendritic cells are act as a antigen presenting cells. Okay, and has a capable to phagocytosis. And next cell is that lymphoid. Next cells of these are the cells actively involving in the innate immunity. And we have another cells. Eosinophils are we have discussed eosinophils, neutrophils, natural killer cells. Okay, natural killer cells or large granule lymphocytes are the cells which comes under innate immunity, and it kills viral infected cells. Natural killer cells kills the viral infected cells, and perforins. Okay, perforins and interferon gamma of the or the different types of the cytokines or enzymes releases from the natural killer cells in the time of the infection, and it kills the, it has a capable to kill the viral infected cell. Especially it also activates the T cell mediated immunity. Okay, T cell mediated immunity can be activated by the natural killer cell. Another cell is that platelets. Okay, platelets, erythrocytes also comes under innate immunity, and platelets releases the mediators in presence to the parasitic infection. If any parasites infection is there, platelets releases the some of the mediators. Okay, which activates the complement system. Platelets releases the mediators whenever the infection is there in our body, and this mediators goes and activates the complement protein. Thereby, the alternate pathway or classical pathway of the complement system can be activated and clears the infection area or site or tissue or foreign body. And what is the importance of the erythrocytes in the innate branch? It binds with the small immune complexes. Okay, these immune erythrocytes goes and binds with the small immune complexes help to remove from cellular circulation. Okay, especially these red blood cells binds with the immune complexes wherever the antigen antibody complexes are there. Sometimes, except those some specific times, it recognizes the immune complexes and it helps to remove the immune complexes from the circulation. So these are the cells actually involved in the uh, innate branch. Understand? Understand, Amma? Hello? Hello?
Yes, sir.
Okay, Namma. This is about innate immune cells.
Okay, sir.
Next, coming to the adaptive cells of adaptive immunity. Okay, these in this branch, in this branch, only two types of cells are actively involved. Those are B lymphocytes and T lymphocytes. Okay, these cells comes from the lymphoid progenitor cell, exactly from the hematopoietic stem cell, and these lymphoid progenitor lymphoid cells and T cells, and B cells. The T cells, in the T cells, we have three types of cells: TC cell, TH cell, S cell. B cells, especially produced in the bone marrow, okay, from lymphoid progenitor tissue or cells, and protects against the extracellular microbes. These cells protects against extracellular microbes, not intracellular microbes, extracellular microbes. From the extracellular microbes, B cell will protect. And B cell, whenever this B cell get activated, it gives two types of cell memory. Plasma cell, memory cell. Plasma cell is a factory or manufacturing site of the antibodies. Okay, plasma cell can also be called as manufacturing uh, site of antibodies. Okay, bursa fabricus in the birds is a manufacture site for the or maturation site for the B cell based upon the maturation. Okay, bursa fabricis is the site for in the bird. In the man, in the human being, it produced in the bone marrow. Okay, and coming to the T cell, T lymphocytes also produced in the bone marrow and activate or mature in the thymus. It protects from the intracellular microbes. See, B cells always protects from the extracellular microbes. T cells protects from the intracellular microbes. That is the difference. That's why B cells actively involving in the humoral immunity. T cells actively involving in the cell-mediated immunity. T cell receptors are there on the surface of the cells. For example, CD4, the clustral differentiation molecule. The meaning of the CD means that clustral differentiation molecule. 4, clustral differentiation molecule 8. Based upon these molecule, we can differentiate from one cell to another cell. That is a TH cell to TC cell. Always this CD4 is there on the TH cell. Coming to the TC cell, always the CD8 present on the TC cell. And CD4 molecule always binds with the MSC molecule 2. CD8 molecule always bind with the MSC molecule 1. Okay, and TH cell activate B cells by the releasing the signaling molecule, bind with the MHC molecule 2. You know about the entire things we have discussed about cell in the cell-mediated immunity just before. And coming to the TC cell, it eliminates the cells. Those are displaying the foreign antigen complex with MHC molecule 1. MHC molecule 1, wherever the MHC molecule 1 is there on the surface of the cell, that is our own body cell, infected cell, this TC cell go and bind with the MSC molecule 1. By the binding By this way, it eliminates the cell, it eliminates the infection, especially viral infection. Okay, bind with the MSC molecule 1 by CD8 molecule, especially in case of viral infection. Okay, virally infected cells can be eliminated or killed by the TC cells by cell mediated immunity. This is about cells involving in the immune system. Cells involved in the immune system. Two types of cells are there: the innate immune system cells, the adaptive immune system cells. Clear?
Yes,
Okay,
sir.
Namo. Okay. Now I would like to talk about lymphoid organs. Okay. Coming to the lymphoid organs, we have two types of in our system. Two types of lymphoid organs are there. One is that primary lymphoid organ. Second one is that secondary lymphoid organ. Okay. What is the meaning of primary lymphoid? Primary lymphoid means lymphoid cells are formed and matured. Where exactly the site? Based upon the site, based upon the formation and maturation. Okay, where exactly the all lymphoid cells formed and matures. Those organs are called as primary lymphoid organs. For example, we have two primary lymphoid organs. One is that bone marrow. Bone marrow is the primary lymphoid organ, and second one is that thymus gland is another lymphoid organ, where exactly the lymphoid cells formed and matures. Okay, coming to the bone marrow, this is the main site of all types of circulating blood cells. Okay, bone marrow, where exactly all types of cells are manufactured, and in the fetus, hematopoiesis takes place in the yolk sac. Okay, in the fetal cell, fetus fetal liver cell and spleen cells releases the will give the different types of will the function of bone marrow replaced by the in the stage of the fetus. This bone marrow function will be taken by the yolk sac and liver and spleen in the fetal age, and antigen independent antigen independent action will be there in the bone marrow. B cell maturation and proliferation can be seen in the bone marrow especially. That's why we can call it as bone marrow is the primary lymphoid organ. Don't bother about the diagrams. Just see the diagram. You can understand the everything about the diagram. From the school time, we have been learning about the bone, TS, and etc. etc. Just know about the function. Why we calling about uh, why we calling primary lymphoid? What is the exactly meaning of the primary lymphoid organ? Why the bone marrow is calling as uh, primary lymphoid? That is the major point. Coming to the second organ is that thymus gland. The development from endoderm, developed from the endoderm in the mediastinum, the thoracic cavity. Thymus gland is there, is present just above the in front of the heart, and behind the sternum. The thymus gland is there. The thymus can be divided into two areas: cortex region. One is that, another one is that medulla region. In the cortex region, we can found immature T cells. Immature T cells are there in the cortex region, and mature T cells with CD4 mark, CD4 receptor. Immature means in the immature stage, the CD4 receptors are not attached on the surface of the T cell. We can also, that's why we call as the immature stage. Whenever the CD4 receptors are attached to the TH uh, cell surface, we can call it as mature cell. Okay, four types of cells are there. The cells are there in the thymus gland: thymocytes, dendritic cells, epithelial cells, macrophage cells are there in the thymus gland. Okay, thymus, in the thymus gland, especially medulla region. Okay, especially in the medulla region, four types of cells are there: matured thymocytes, dendritic cells also there in the thymus gland, and dendritic epithelial cells, macrophages are migrate and reside, store in the medulla region, uh, exactly waiting for the encounter. Okay, this is about the uh, two primary organs. One is that bone marrow, another one is that thymus gland. And we have secondary lymphoid cells, sorry, organs. What are the secondary lymphoid organs? Where exactly maintenance of the it maintains the mature nerve lymphocytes. It maintains where exactly maintenance and site of the activation, not maturation, but activation of the lymphocyte cells will be taking place in the secondary lymphoid organs. Okay, mature lymphocytes encounters by the antigens and activation of the lymphoid cells can be taking place in this type of organ. Understand, Amma, the difference between two organs, but exactly difference. Okay. Hello.
Yes, sir.
Main difference is that primary lymphoid in the primary lymphoid organs in which lymphocyte cells are formed and mature. That is about primary lymphoid cells. What is there in the secondary lymphoid, in case of secondary lymphoid organs, in which mature cells encountered by the antigens and get activated in the secondary lymphoid organs. For example, lymph nodes. Lymph nodes are the secondary lymphoid organs where exactly activation of the lymphoid cells taking place. Encounter fighting between the antigen and immune lymphoid cells taking place in these organs, not primary organs. And the second one is that spleen. Okay, spleen is also a secondary lymphoid organ. In the spleen, the regions, three regions are there: red pulp, white pulp, cross section. If we cross section the spleen, we can find three areas: red pulp, white pulp, marginal zone. In the red pulp, macrophages are there, erythrocytes are there, filtered blood borne antigens are there. What is there in the white pulp? In the white pulp, arteries rich in T cells, dendritic cells are there in the white pulp region. In the marginal zone, rich in B cells. Okay, these spleens in the spleen marginal zone rich in enriched by the B cells and encounter with the antigens. Okay, and coming to the other secondary lymphoid organ is that mucosal associated lymphoid tissue. Okay, mucosal associated lymphoid tissue, malt. Malt also can also act as a secondary lymphoid organ. We have different types of mucosal associated cells and rich in macrophages and designed to facilitate the antigen trapping. These are the sites where exactly antigens can be trapped. Enhanced phagocytosis pathway and malt. Coming to the malt, tonsils. Okay, mucosal associated lymphoid tissue is there in the tonsil area. Pairs, patches. Okay, adenoids. Appendix also contains malt and mucosal membranes and different types of mucosal membranes. Okay, and enriched with the different types of immunogen antibodies. Antibodies A. Okay, these are the secondary lymphoid sites. And fourth one is that uh, different types of areas are there. Uh, Nasal associated lymphoid tissue, gastrointestinal associated lymphoid tissue. Okay, different types of lymphoid associated areas are there. Those are also acts as a secondary lymphoid organs. Okay, this is about types of organs. We have two types of lymphoid organs. One is that primary lymphoid organ, secondary lymphoid organ, and this is about the organ. And I would like to talk about antibodies. Okay, antibodies are the magic bullets. Okay, antibodies also called as a magic bullets. Just wait for a minute. I will show one image for you. Yes, antibodies can also called as magic bullets of immune system. These are the glycoprotein molecule or globular proteins secreted or synthesized against an antigens from effector B cells, and the part of the antibody that recognizes on antigen is that paratope. Paratope, epitope is a very, very important terminology. Paratope is the site there in the antibody where exactly antigen, where exactly antibody communicates with the antigen. Paratope is a site or part of antibody where exactly this part, where exactly this antibody communicates with the antigen. That those sites are called as paratopes or paratope. And coming to the epitope, what is epitope? This is the active site on the antigen. Active site of the antigen is called as epitope, where exactly the antigen binds with the paratope. Epitope is the antigen part, paratope is the antibody part. This is the main difference. Based upon the mobility, antibodies are two types: circulating antibodies, surface antibodies. Two types of antibodies are there based upon the mobility. What are the circulating antibodies? Circulating antibodies circulates in the blood body fluids, that may be lymphoid, lymph or blood. Uh, Surface antibodies are always present on the memory cell. Okay, memory B cell. Uh, Whenever the B cell get activated, it gives the uh, memory cell. Okay, and especially coming to the structure, Y-shaped structure is there for the almost all Y-shaped structure is there for the antibody proposed by the Rodney Porter. Okay, four polypeptide chains are there, two heavy chains, like two light chain polypeptide chains. Two chains are linked with the covalent disulfide bonds, beta disulfide bond. Important, very very important. Disulfide bonds are there in between chains. Okay, two ends of antibody chains, that carboxyl end, COO end, NH4MN end. Two ends are there. It has two regions. Okay, antibody contains two regions. One is that Fab region. Okay, upper one is that, for example, I will draw a diagram Y shape. This is the Fab region, and this is the FC region. Okay, from here to here, we can call it as a Fab region. Fragment antigen binding. Okay, fragment antigen binding site. Fab full form is that fragment antigen binding site. FC region means fragment crystallizable. Okay, FC means fragment crystallizable. Okay, now to fragment cell binding region. So cell binding region where from the macrophages, from the neutrophils to the natural killing cell, or to the B cell, different types of cell where exactly the antibody is attached with the help of FC region. Okay, with the with the help of FC region, it binds with the cells surface. Okay, fragment cell binding region, the lower one. Upper one is that fragment antigen binding region. Okay, dominant domains, not dominant domains. Two domains are there: heavy domain, light domain. Heavy domain means VH. Okay, variable region, heavy chain variable region, constant heavy region. Okay, you can study all those things. Okay, and light chain, in the light chain, different types of molecules are there. Kappa region, kappa, and lambda chains are there. And heavy, coming to the heavy chains, different types of heavy chains are there. 1, 2, 3, 4, 5. Okay, based upon the heavy chain present in the antibody, we can name it as a different. We can give different types of names. If the antibody contains a heavy chain, we can call it as immunoglobulin A. If the antibody contains mu chain, immune antibody called as M, immunoglobulin M. If the heavy chain is epsilon contains epsilon heavy chain, if the antibody contains epsilon heavy chain, we can call it as immunoglobulin E. If antibody contains gamma, okay, we can call it as immunoglobulin G. Based upon the heavy chain, we can call name can be give. Okay, name can be given as based upon the uh, name of the heavy chain. Okay. Coming to the immunoglobulin function or antibody function, three major functions are there. One is that neutralization of antigens. Okay, antigens neutralization can be the one of the function, and punch holes or make pores on the cell wall of microbes. Whenever these antibodies goes and binds with the foreign or infected cells, this uh, binding facilitates the attraction of the different types of classical uh, pathway proteins, alternate pathway proteins. Okay, and by after that, pores pores can be the consequence of the attraction, alternate pathway, classical pathway uh, may leads to the pore formation on the site. Okay, the infected site. Due to this function, due to the attachment of the antibody, enhances the attraction of the different types of the proteins. And another function is that opsonization. Okay, third function is that opsonization. What is opsonization? It attracts the or activates the phagocytosis mechanism. Okay, phagocytosis mechanism can be activated by the attachment of the antibodies on the surface of macrophage or dendritic cell. After attachment of these antibodies, phagocytosis can be activated, and this is a opsonization. Okay, it helps to phagocytosis pathway process, and these are the function: neutralization, punch holes. Opsonization is the major function of the antibodies, and specificity is the diversity, immunological memory, immune tolerance is the with different features of antibodies. Okay, uh, uh, exactly. I am going to stop at this point. So we will talk about the classes uh, of antibodies, different classes of antibodies tomorrow. After talking about this, I will enter into the new topic. I will introduce new topic. Okay.
Okay, sir.
Okay, Namma. Okay. I am going to leave. If any doubts are there, you can ask tomorrow. Okay, you will ask. You can ask in the tomorrow first session, first 10 minutes. Okay.
Okay, sir. Thank you.
Okay. Okay, ma'am. Right. All right.